Hello Biology 223 and welcome to Snowpocalypse 2017, or more like Slushpocalypse 2017. As of right now the weather is not too bad. So I am recording our Chapter 18 Autonomic Nervous System lecture right now because once you've covered Chapter 14, I think that Chapter 18 can stand alone as long as you know what things like synapses are, what ganglia are. You should be fine to go over this early. The other reason I chose to do our last week lecture is that it's going to work out best for everybody, no matter what our scenario is going to be. I'm recording this in the morning. I don't know yet if we're going to have a makeup day. If we don't, then you're going to need to make up our last week lecture, so this works for you. And if we do get a makeup day, maybe you won't be available for our makeup day. Maybe you'll be at work that day. And even if we do get a makeup day and you get to go to it, that only leaves you with a couple of days before our final to assimilate all of this autonomic nervous system information. So it's really not going to hurt for you to go through this early. May have a little bit more time with it, and I think I will upload that worksheet early as well so that you can work through it and have plenty of time to ask questions. We'll have plenty of class time to review because I'm casting this lecture. Hopefully this helps the vast majority of you. So we've covered the general divisions so far in Chapter 14. You know that there is motor and sensory divisions of the functional organization of the nervous system, and within those divisions you also have a sensory and motor, and sensory you have an autonomic division, or you have a visceral division I should say, and in your motor you have an autonomic division. So we're going to compare somatic versus autonomic one more time. Divisions of the autonomic nervous system, it keeps dividing. Parasympathetic and sympathetic. It's really not as much of a division as you shouldn't worry about it just yet. Alright, parasympathetic is going to be rest and digest. Sympathetic is going to be fight or flight. Other features of the autonomic nervous system and how the central nervous system controls autonomic functions. Now there is a really good crash course on this. Let me move this screen around for you. So these are really fantastic, and generally when we do have class time, we spend some class time looking over the crash courses for these, but again, he's a really fast talker. Hopefully I'm a little bit of a slower talker than he is. So one thing you might want to do is go to YouTube and look at the overview autonomic nervous system crash course once, and then come back to our lecture, listen to the lecture, and then go back and review the crash courses for those. That's a really good strategy for this system. Never hurts to have a second person giving you advice. Since this is a screencast, I'm not going to, have that class time to show you crash course during class time. So the somatic nervous system, you should already know, is voluntary and generally speaking conscious. Right? We're going to have a sensory receptor in the skin. It's going to travel to the central nervous system, and there's going to be a motor neuron that is an effector that travels from the CNS to an effector such as a skeletal muscle. So that's somatic. There's not necessarily conscious decision making. For example, this is actually demonstrating a reflex arc. So there was no conscious decision in this, but it was somatic because it is part of our voluntary system. Autonomic, you can pretty much just go ahead and say automatic. I maybe had some teachers get angry at me for saying that in undergrad, but you know what? It works. It's all automatic. We don't have to have any conscious thought, any conscious decision making. It all travels through a slightly adjusted system to do things for your body without you having to think or worry about them. So if we had to think about breathing, if we had to think about heart rate, if we had to think about where we're shunting blood in our bodies, we would never get anything done. We'd be sitting there just trying to maintain our basic bodily functions. So our autonomic nervous system takes over those functions for us so that we don't have to worry about them. So autonomic nervous system includes processes regulated below the conscious level. There are actually some diseases. There's at least one disease, can't remember what it's called right this moment, but your breathing is not regulated automatically, so you have to consciously sit there and breathe, and if you ever forget, then you'll just stop breathing. I've actually seen a person with that condition. Both of these divisions, somatic and autonomic, use sensory and motor neurons. It's still the same organization. They're still afferents, they're still efferents. They're just going to be conscious versus unconscious. We're going to still need effectors for the autonomic nervous system. In the autonomic nervous system, the difference is that it's going to innervate smooth muscle, cardiac muscle, and glands. So those are the things that we can control autonomically. So again, glands, we'll start talking about at the beginning of ANP2, are about hormones, or secretions in some cases, but for our purposes today with autonomic nervous system, we're going to think of glands as being hormonal. So hormones are going to interact with the central nervous system to control bodily functions and our state and our arousal level, our consciousness level. Hormones and neurons work together. And again, those motor neurons can be excitatory or inhibitory, so increasing the action of something downstream or decreasing the action of something downstream of an effector. I love this image of a peripheral nerve. So I'm just kind of bringing you back to this concept of the peripheral nerve having both sensory and motor. It's going to have both somatic and autonomic neurons traveling together in the periphery as well. So remember, a peripheral nerve contains all of the divisions that we've talked about in our functional organization of the nervous system. They all travel together once they are out of the central nervous system. So that is what this is portraying. So in our somatic nervous system, we only had one lower motor neuron. As of our lecture in this quarter, we have not seen those pathways, so you have not seen the upper and lower motor neuron pathway. It's known as the corticospinal tract. That's all you're really going to learn about the motor pathway, is that there's an upper and a lower. So we know that there's just one lower motor neuron in our somatic nervous system to control muscles. In the autonomic nervous system, you have two neuron chains. This two neuron chain is going to be our organization that divides sympathetic with parasympathetic. So this is going to be our basic division, is how do those two neurons look? They are much more significant in the autonomic nervous system in terms of our organization and how we're going to think about it than our pathway in the somatic nervous system. We are going to focus a lot on what is the shape and form of the preganglionic neuron and what is the shape and form of the postganglionic neurons. This should really say post right here. Postganglionic neurons. Here's what we mean by this. So recall, we're going to be assembling this list. What things are cell bodies and what things are axons? A ganglion is a collection of cell bodies in the peripheral nervous system. So on our running list that we started at chapter 14, ganglia goes under the cell body section. And then obviously you have axons, and those are axons. These will travel within our peripheral nerves along with every other division of the nervous system. So for our autonomic nervous system, we're going to have a preganglionic neuron that synapses in a ganglion, and then a postganglionic neuron that synapses onto an effector organ. So cardiac muscle, smooth muscle. In this example, we're using smooth muscle that surrounds all of your small intestine. So remember, we need to have a system for rest and digest, right? So this is going to be part of our rest and digest system that's going to increase digestion when we are not running away from bears. We'll talk about that more in a bit. I always use the running away from bear, running away from the tiger, because it's nice and extreme, and it's going to make us... using the extreme examples helps you to familiarize yourself with the general concept. It helps you categorize what the effects of each are going to be. Okay, comparison of somatic versus motor. Really, we've talked about the important stuff. The effector organs being conscious skeletal muscle versus involuntary cardiac smooth muscles plus glands in that category. And that should look really familiar to you, right? We've already talked about our muscle fiber types and whether they are voluntary or involuntary. Now we're just giving you a name to that system that is controlling the involuntary stuff. So parasympathetic division we consider our rest and digest division. What this means is you are not currently in fight or flight. You are going to prioritize things when you are in different states. If you are in fight or flight, if you are, fighting a person, fighting a bear, running away from a person, running away from a bear, you are going to need increased alertness, increased metabolism, increased heart rate, increased respiration, and increased blood flow to very specific regions. In this case, it's going to be the peripheral skeletal muscle. Those muscles that you're going to use to run away or fight somebody. So that's our sympathetic division. Then our parasympathetic is when we're not doing that. Then we can devote our energy to digesting nutrients, processing things. Digesting your breakfast, not super important when you're fighting a bear. Digesting your breakfast is super important when you're not fighting a bear, because that's when your digestive system can take those nutrients and process them, and guess what? Use them for everything that is super important for building the cells of your body. So they're both equally important. There's not one that's more important than the other. Most significant for these divisions, we give you those extreme examples. We give you running from a bear or a person, and we give you chillaxing on the beach, right? Completely different scenarios. The truth is, it's more like a seesaw. It's more like every moment of every day, your rest and digest system is working with your fight or flight system to give you the correct level of excitation, arousal, in any given scenario. So if you're sitting here listening to this, you should be relatively resty and digesty, but you also need a certain degree of awareness in order to absorb what I'm telling you, right? So that it's really a balance. It's very rare when you're actually running from a bear, but on, for example, a test day, that's a kind of high anxiety thing. Your sympathetic division hormones and neurotransmitters may be a little bit higher compared to it's a snow day and I'm sitting at home with a coffee, right? So they're always balancing each other out. We give you the extreme examples because it makes it easier to understand the patterns, but just understand that they're always both working in conjunction to give you the right level of consciousness, alertness, metabolic activities, respiration rate for what you are doing. All right, so this is going to be, if you're trying to fit everything from this lecture onto one piece of paper, and you absolutely can, here's going to be the first part. The parasympathetic division is cranial and sacral. The sympathetic division has its roots in the thoracic region, from T1 to L2. This is just the beginning concept. Have you ever heard of craniosacral therapy, for example? It's a field that exists in massage therapy. I'm not super into it, but it's the idea of utilizing your parasympathetic nervous system to relax and adjust things to allow for some other things. Don't worry too much about that technique. Just think of it as relaxing, right? Craniosacral massage is relaxing. So that's how you're going to remember that parasympathetic is in the cranial region and in the sacral region. Sympathetic division is from the thoracic region. This is also going to be on your one sheet of paper that can sum up this entire lecture. In the parasympathetic division, preganglionic neurons are long and postganglionic neurons are short. In the sympathetic division, preganglionic neurons are short and postganglionic neurons are long. There is a functional reason for this, and we'll get into it, But in terms of memorizing short, long, long, short, don't overthink it. Guess what? Parasympathetic division, preganglionic neuron is long, postganglionic neuron is short. Sympathetic division, preganglionic neuron is short, postganglionic neuron is long. That's really all there is to it at this point. The only way that this is going to get more complicated is when we talk about the ganglia of the sympathetic division. So the ganglia of the sympathetic division is why the preganglionic neuron is short, and there's a functional correlation for the location of the autonomic sympathetic ganglion. Again, all you need to know, long, short, short, long. That's it. Full stop. Don't overthink it. So I don't want to overly confuse you guys, but there has been a sort of scientific breakthrough just in the last year or so about the sacral division of that craniosacral part of parasympathetic we were just talking about. I just told you that your neurons of the parasympathetic division arise from the cranial region and the sacral region. And I told you to think of craniosacral massage. In actual reality, in the last couple of years, we've been discovering that the sacral outflow is actually sympathetic. For our purposes, the roles and structure are going to be the same, though. So the reason we thought that sacral division was parasympathetic was because it had these rest and digest functions, right? And it had a rest and digest pattern of, wrong way, long preganglionic, short postganglionic, right? It shared those patterns, but we're finding that the neurotransmitters involved in those inhibitory, regulatory, metabolic actions are more similar to the sympathetic nervous system and are moderated by the sympathetic nervous system. So you don't have to memorize that right this second. For our purposes, you can just say that the parasympathetic division is derived from the craniosacral regions. I just want you to be up to date on the science, which is why this is here at all. I think it's super cool that we're rethinking sacral outflow. From a neuroscience perspective, it's huge, it's awesome. From a student perspective, I'm just trying to memorize this and pass a test. Maybe not as exciting, so I get that. Ganglionic neurons of the parasympathetic division will be found close to or on or in the target organ. So I mentioned up here there's a functional correlation with the location of ganglia for the sympathetic division. For the parasympathetic division, those ganglia are going to be all over. So if it's going to your small intestine, you might have a ganglion embedded inside the wall of your small intestine, and then that super short postganglionic axon is going just right to the immediate area, right? It's going all throughout your digestive system. So that also has a functional correlate. All of these ganglia are going to be very far away from each other, and that's going to mean that it's harder for one parasympathetic neuron to communicate with another pathway of the parasympathetic division. So there's less communication between the parasympathetic neurons compared to different sympathetic neurons communicating to each other because of this arrangement. And again, it's more about the significance of the ganglia in the sympathetic division. In ANP2, we are going to talk about the vagus nerve all the time. If you can remember the term vagus nerve throughout ANP2, almost every single time I say, okay, what do we think this organ is innervated by in the parasympathetic division? If you say vagus, you're going to sound like a rock star, and it's almost always going to be vagus, because here's our ANP2 class. We do cardiovascular in ANP2, we do respiratory, we do digestive, we do urinary, we do reproductive, which is the only exception here. Well, urinary and reproductive are the only exception to the vagus nerve innervates almost all of your viscera for the parasympathetic pathways. So your rest and digest system is almost entirely modulated by one cranial nerve known as the vagus nerve. So your vagus nerve for parasympathetic, and remember, we're talking rest and digest. So if you're just sitting still, you're just listening to this lecture, you maybe just ate an apple, do you need your heart rate up or down? Probably down. You do not need a high heart rate if you're just sitting here. If you have a high heart rate when you're just sitting here, it's actually a bad sign. It means something's wrong, right? We don't like high heart rates at rest. So your parasympathetic nervous system, your vagus nerve, slows down your heart rate. It also travels to your lungs, and if you're just sitting here, not doing much, do you need to be breathing very heavily? Do you need a lot of airflow? No. You need little airflow. You just have this small tidal volume of air you're breathing and inhaling and exhaling. So it's actually correlated with what we call bronchoconstriction. So decreasing the width of the airways in the lungs is what happens via parasympathetic innervation to the lungs. And then if you're just sitting here and you just had an apple, do we need to increase blood flow and activity to the digestive system? Yes, we do. So it's actually going to increase movement along the digestive tract, increase blood flow to your digestive organs, increase activity of your liver and kidneys, and even to an extent your kidneys, your spleen, your pancreas. Your parasympathetic nervous system increases the action of your digestive organs. That's why we call it rest and digest. These are resting, these are digesting. And again, sacral outflow has been rethought. These systems may actually be modulated via the sympathetic nervous system, but for our purposes, if you want to just stick with cranio and sacral being parasympathetic outflow, go for it. It's okay for now. Let's focus in for a second on what we mean by cranial. What we mean by cranial is coming from the brainstem, cranial nerves. So as of this current lecture, as of this date, we haven't gone over what all of our cranial nerves are doing. It's not super important for you right now. So cranial nerve 3, we will learn, controls extraocular muscles. For the parasympathetic division, it's also going to control lacrimation. Then your facial nerve is going to do muscles of facial expression. It's also going to do salivation via salivary glands. So recall that our target organs for the autonomic nervous system includes glands. So lacrimal gland via 3 and salivary glands via 7. The rest of it is vagus. Everything else is vagus. What happens in Vegas does not stay in Vegas. It goes everywhere. Vegas, the name Vegas actually means wanderer. And you can see why it means wanderer. It's going everywhere. So oculomotor is cranial nerve 3, facial is cranial nerve 7, glossopharyngeal is cranial nerve 9, and vagus is cranial nerve 10. Let's see, glossopharyngeal otic ganglion. I think that does one of our salivary glands as well, if I'm recalling correctly. Pupillary constrictors and dilators are also moderated via the autonomic nervous system. So pupillary constrictors are going to be controlled via also oculomotor nerve, cranial nerve 3, in addition to your lacrimation, crying. That parasympathetic stimulation is also going to constrict pupils. It can seem counterintuitive that relaxation, resting and digestion, is going to correlate with a constricted pupil size. The way that I think of it is more about the sympathetic division. If you are in fight or flight, what happens to your pupils? What happens when you put a cat toy in front of a cat? Think of the pupils of a cat when it's really excited. They get huge, right? There's your cat's pupils dilating hugely because the sympathetic nervous system has taken over in your cat. It's in fight or flight. It's ready to fight that toy mouse in front of it. So by definition, if the sympathetic division of the autonomic nervous system is causing pupillary dilation, then by definition the parasympathetic is going to give you dilation. When you're in rest and digest, your pupils are more likely to be dilated. Again, this goes right back to that concept of, we talk about these in the extremes of fight or flight and rest and digest, fighting a bear, sitting down, reading a book, right? But you need to dilate your pupils or constrict your pupils in other scenarios, right? It's about how bright light is, your response to changes in light. It's about how close the book that you're reading is to your face. So again, we talk in terms of these extreme scenarios, but your autonomic nervous system is working all of the time to adjust these things at a fine-tuned level for you. So your sympathetic nervous system is dilating your pupils when you're in the dark, even though you're not necessarily running from a bear in that scenario. It's still just controlled by that portion of the nervous system. That's all. Facial nerve to, let's see, I was wrong about lacrimal. So lacrimal is not from 3, lacrimal is from 7. Crying is from 7. Submandibular and sublingual salivary glands. You'll learn about those during digestive lecture in ANP2, but you have saliva. You know you have saliva, so you have glands that secrete saliva, and that's controlled via your facial nerve. Glossopharyngeal does your parotid salivary gland. So 3 different salivary glands. The parotid is just anterior to your ear, and that one's going to be controlled via glossopharyngeal. And then vagus, our wanderer, and I really like this image because that vagus is in yellow. It just goes everywhere, right? It's all over the place. So again, ANP2, what parasympathetic nerve innervates this organ? Go ahead and guess vagus. You'll look awesome 90% of the time. And again, that's going to increase smooth muscle motility and secretory activity in the digestive tract organs. So that's why we call it rest and digest. We're slowing down respiration, we're slowing down heart rate, but we are increasing activity in your digestive organs in comparison. You also have pelvic splanchnic nerves. So these are going to be those sacral division nerves that control your pelvic organs for the most part. Let's see. Your part of your large intestine, your rectum, reproductive organs, urinary bladder, and distal ureter. So kidney was back with vagus, but urinary bladder and ureters, your actual urination as a process, goes through pelvic splanchnic nerves. And we divide those from that vagus. Again, it's a little bit about the organization of their ganglia. So sympathetic division, fight or flight, also termed thoracolumbar because it comes from T1 to L2. So the entire thoracic spinal cord all the way down to the L2 level of the spinal cord. And again, we haven't gone over our shape and form of the spinal cord as of yet. All you need to know is that you have this butterfly shape. This is gray matter of the spinal cord. And I've already shown you ventral versus dorsal divisions. This is going to be mostly about sensory stuff. This is going to be mostly about motor. In that thoracic region, T1 to L2, we're also going to have a little bit of gray matter over here called the lateral gray horn. I do care about the word lateral gray horn and its location, which is right here. You are only going to have a lateral gray horn from the T1 to L2 spinal cord segments, because this is where your preganglionic neuron cell bodies are going to originate. Preganglionic axons travel with the somatic motor neuron, so this is our motor division. So cell bodies for motor start here, cell bodies for sympathetic start here, and they both have this ventral outflow, and then they join up into a spinal nerve with the sensory division from up here. I promised you there would be a nifty organizational thing related to the ganglia of the sympathetic pathways. So here is something called the sympathetic chain ganglia. Every one of these little bulges represents a ganglion here. And they are connected to one another in a chain. It's the sympathetic chain of ganglia. So recall that in our sympathetic pathways, the preganglionic neuron is short and the postganglionic neuron is long. Preganglionic short, postganglionic long. Preganglionic short, postganglionic long. This is why that preganglionic neuron tends to be short, because it goes straight to that sympathetic chain and then synapses. And something that it can do in the sympathetic chain is it can ascend to higher levels, or it can descend to lower levels, so that its outflow can reach all over the body. Recall, parasympathetic had cranial and sacral divisions, and it had to descend via the vagus nerve to get to all of these, and since it was in control of several cranial nerves, or traveled along with several cranial nerves, it was already in the cranium and it could get to the face just fine. If you are in the sympathetic pathway, you still have to innervate parts of the cranium. You still have to do that pupillary dilation, for example. So that's going to be an ascending pathway. Those neuron cell bodies still come from the thoracolumbar region, T1 to L2, but they have to ascend through the sympathetic chain to get up to the head. Same thing for down here. We don't have, well, we do, but historically we considered it to nothing below L2 being sympathetic, right? So then we have the sympathetic chain, and those preganglionic neurons simply travel down the chain before synapsing and having their outflow to the pelvic organs that need sympathetic innervation. So does that make sense? You have this long chain, and the purpose of it is partially about ascension and descension, ascending and descending pathways. It's also about communication between these two things. If you're running from a bear, you are 100% running from a bear. All systems go. You need dilated pupils so that you can see the bear. You need a fast heart rate so that you can get oxygen to everything that needs it. You need dilated bronchi in your lungs so that you can get more air. And we're going to shut down those digestive organs because we do not need them right now. We need to be running away from a bear. So everything happens at the same time, because aside from ascension and descension, this is also about communication. It is way faster to get into fight or flight than it is to come down from fight or flight and into rest and digest, right? So you have this intense communication in the sympathetic pathway. Whereas in the parasympathetic pathway, you have your pupils doing one thing, your saliva doing another thing, your pelvic organs doing a different thing. And then, you know, your viscera in general, they can do their own thing via vagus because they're all controlled together. Once, you know you're not running from a bear, it's going to take some time for your heart rate to come down and for your breathing to level out. And it's going to take some time for your digestive activity to come back, because they don't have their ganglia communicating with each other in any direct way. It has to come back through circuitry into your brain. You have to consciously be telling yourself, I'm not running from a bear, I'm okay. And you know, also your autonomic divisions are gradually enforcing and gradually sending signals throughout your body that you're in rest and digest again. You no longer have to freak out and be in fight or flight. But the moment another bear shows up, zoom all along that sympathetic chain there. All systems are go, we're ready to run from the bear again. That has a big contribution to stress responses, right? So you had an exam in the morning and you're kind of stressed all day, right? it can take a while, or it can take conscious effort to hijack your parasympathetic nervous system to bring you back down from a stressful event. So if you're a person who has panic attacks or anxiety, there are actually things you can do to learn how to control your parasympathetic nervous system. If I can, I'll have my friend guest lecture again about just how to do that. It's a really interesting lecture. So this is just talking about your sympathetic chain ganglia. I prefer sympathetic chain ganglia. It's what I was taught. I think your book uses the term sympathetic trunk more frequently. It's immediately lateral to the vertebral column. It's very close to your spinal cord. Hence the short preganglionic neuron. Short preganglionic neuron in the sympathetic division. Here's a beautiful dissection of the thoracic region. So these are the bodies of your vertebrae. You have your thoracic aorta and a major vein of the thoracic cavity. And they've removed the heart, they've removed the lung, and they've removed a membrane called the pleural membrane to show you this pathway right here. So that's a ganglion, that's a ganglion, that's a ganglion, that's a ganglion, and they're all connected to one each other. These are all nerves coming down from it, right? And you have a nerve pathways going through each of your ribs. We're also going to learn about the white ramus and the gray ramus. So you kind of have to zoom way in to see where these are. There's the white ramus, there's the gray ramus, a little bit more medial in this image. It's a gorgeous image. Your sympathetic pathways vary based on targets. Your adrenal medulla has its very own, very direct pathway. So if you're looking at this early and you've never seen a pathway before, pathways can be a little bit confusing. I like to think of it as learning a subway station, right? So you get on the subway at the lateral gray horn, and that takes you on the ventral route. You make it to the spinal nerve, and then it takes a sharp right down the gray ramus, and it synapses. So when we synapse, we're saying you get off one train and you get on another. So you go stop at the train station, you get off, you get onto the postganglionic neuron, the second train in your pathway, and that's going to take you to the place you actually need to go. It's going to exit via the gray ramus, and it takes you where you need to go. So it's like a subway station. Learning a new subway station in a new city, but all it is a pathway. I do have this. So it's pretty much the same thing as, alright, I'm getting on the red line at the Loyola station, but I need to get out to, let's say, Ridgeland in Chicago. This is the L. This is Chicago, in case you've ever been to Chicago. So I get on at the Loyola station and I go south. And then I have to get off at Grand, and I have to walk a few blocks over to the Green Line and find a Green Line station. Let's say I get back on at Clinton, and then I head west until I make it to Ridgeland. It's the exact same process. It's just you use neuroscience terminology now. So starting at the lateral gray horn in vertebral levels T1 to L2, you exit via the ventral ramus, join up with the spinal nerve, go in via the white ramus to the sympathetic chain, and then it synapses onto the postganglionic cell body. The postganglionic cell exits via the gray ramus and heads all the way to the target organ. That's for blood vessels. I believe that's going to affect smooth muscle, things like that. Different things will have slightly different pathways. What we mean by white ramus and gray ramus is that the white ramus is myelinated and the gray ramus is not. And we've already learned about myelin. So myelin sped things up and insulated. In the gray ramus, things slow down just a little bit. It's also worth noting that when you get to the sympathetic chain, there are some options here. Some of these neurons synapse at the same level of the sympathetic chain and exit to the target organ. Some of these can ascend or descend along the sympathetic chain before synapsing on a postganglionic neuron. So remember, the blue is the postganglionic neuron, the brown is the preganglionic neuron. This is just showing you an ascending pathway through the sympathetic chain. It still comes in via the white ramus, and after the synapse, the postganglionic exits via the gray ramus. Whether it stayed at the same level, or ascended, or descended. And again, remember, this is an all systems on board system, so if this level is excited, then this level is also excited. In this case, the pathway to the heart, the preganglionic cell body entered via the white ramus, ascended or stayed at the same level, or descended, synapses, and then it exits via a different pathway. It doesn't exit via the gray ramus. It goes straight through this nerve right here. It's just a different pathway. For some of your splanchnic nerves, your sympathetic to your intestines, we don't synapse in the sympathetic chain. This can go a little bit more slowly. We can take a little bit more time before we deprive the intestines of oxygen during fight or flight. So it comes in via that white ramus, it exits via a splanchnic nerve, and then there's a prevertebral ganglion. It's a little bit further away from the sympathetic chain. So that preganglionic nerve is a little bit longer. It's still short compared to your parasympathetic nerves. You don't really need to memorize these. You just need to generally understand what's happening. The adrenal medulla has its very own pathway. It doesn't even synapse. There's one sympathetic neuron. It does enter via the white ramus. It exits via splanchnic nerves, goes straight to the adrenal medulla, and that is going to cause the adrenal medulla to release norepinephrine and epinephrine, which are our jazz hands' main sympathetic hormones. Our main sympathetic hormones are norepinephrine and epinephrine. Norepinephrine promotes fight or flight response throughout the body. So hormones, we'll learn later, travel via the bloodstream, and they will affect every single cell that has a receptor for them. So this is another part of our all hands on deck approach to the sympathetic pathways. Once we are in an excited state, we have this intense hormonal release of epinephrine and norepinephrine, and that goes back and affects all of those same organs of the parasympathetic division, sympathetic division. All of these organs will also be impacted by epinephrine and norepinephrine. You also have plexuses, and there will just be a few places in AMP2 where you're going to need to know, okay, the kidneys are controlled by the renal plexus, right? It's going to be collections of sympathetic postganglionic axons and parasympathetic preganglionic axons. Also, visceral sensory axons travel along these autonomic plexuses as well. Why is the sympathetic postganglionic axon traveling with the parasympathetic preganglionic axon? Very simple reason. because the parasympathetic preganglionic neuron was really long. So for the bulk of a spinal nerve or a plexus nerve or any nerve that's traveling in the periphery, it's going to be preganglionic parasympathetic. And in the sympathetic division, the postganglionic neuron is long. So the majority of the axons traveling in autonomic plexuses are going to be postganglionic. It's just because of the length of them. So this portion will be traveling with this portion. That's it. Don't overthink it. So that is why sympathetic and parasympathetic axons, that is why sympathetic postganglionic axons travel with parasympathetic preganglionic axons. If there's any vocabulary that's not making any sense right now, you're going to want to go back to the start and review what that vocab means. So postganglionic means the neuron axon that is after the ganglion. The ganglion is that collection of cell bodies. So you have a cardiac plexus, you have a renal plexus. Mesenteric plexus is going to refer to intestines, so on and so forth. If there's a dedicated slide for it, you're probably going to need to know a little bit about it, but again, they're pretty much named for what they do. The cardiac plexus is in the mediastinum, which we know is that central cavity in the thoracic cavity that contains the heart and things that are superior to the heart. So of course, it's going to impact your heart rate. Parasympathetic decreases heart rate, sympathetic increases heart rate, increased heart rate increases blood pressure. Pulmonary plexus, again, if we're resting and digesting, we don't really need a lot of airflow, so we constrict our bronchi. We have a reduced diameter of our airways during parasympathetic rest and digest innervation. During sympathetic innervation, fight or flight, we experience bronchodilation, which means we have an increased airway diameter. Esophageal plexus, your esophagus is your initial portion of your gut tube, so that's going to allow for coordinated smooth muscle activity during your swallowing reflex as food goes from your mouth to your stomach. And these are going to be, so celiac and mesenteric are going to refer to abdominal organs, mostly the gastrointestinal system. Celiac is also going to include your stomach and your liver and your spleen. Superior mesenteric is going to be mostly small intestine. Inferior mesenteric is going to be a chunk of your large intestine. So innervase most digestive viscera is the way that you can think of abdominal, celiac, superior mesenteric, and inferior mesenteric plexuses. Hypogastric, so we've talked about the hypogastric region before. You know where it is. It's going to be the region that contains your pelvic organs. So innervating viscera within the pelvic region. One more super key thing to put on to your, one-page summary of this entire lecture. We've already got parasympathetic, preganglionic long, postganglionic short, sympathetic, preganglionic short, postganglionic long. The last key piece of information to know about the autonomic nervous system to carry forward with you into AMP2 and pathophysiology is that in the parasympathetic division, preganglionic neurons release acetylcholine onto nicotinic receptors, and postganglionic neurons release acetylcholine onto muscarinic receptors. For our sympathetic pathways, the first neuron is the same, acetylcholine onto nicotinic receptors. The second pathway, the one we're going to be most concerned with, is going to be acetylcholine onto muscarinic receptors. My apologies, that's the exact same thing. This is the pathway we're going to be more concerned with. Norepinephrine onto adrenergic receptors. I should probably redo that, but I'm not going to. So, preganglionic neurons release acetylcholine onto nicotinic receptors. Postganglionic neurons release norepinephrine onto adrenergic receptors. Those muscarinic receptors do have some significance. What does the word nicotine make you think of? Oh wait, I just said it, nicotine. Nicotinic receptors also receive nicotine. When you smoke a cigarette, these receptors are being activated by the cigarette smoke and not by your neurons firing. So it gives you a sort of excitation event along any pathway that has nicotinic receptors. So your autonomic nervous system is altered by cigarette smoking. What about muscarinic receptors? There's a thing here. If you do a Google image search for muscarine, you get a couple things. One of the things you get is this mushroom right here. So if you happen to be out in the world and see a Mario mushroom, don't eat it. It contains muscarine. What's muscarine? Muscarine is going to increase activity of parts of your autonomic nervous system. For our purposes, we're going to be worried about the parasympathetic division right here. Let me fix this real quick. It's not quite filled well. This can depress your system to the point where your heart can stop beating, your lungs can stop breathing, and you can die from eating that mushroom. The poison is in activating these muscarine receptors. So we have a lot of drugs that impact these receptors all along the autonomic nervous system, is what we're getting to. And then, of course, adrenaline. Norepinephrine and epinephrine are the same thing as noradrenaline and adrenaline. Adrenaline and noradrenaline are the same thing as epinephrine and norepinephrine. So when we say norepinephrine is released onto adrenergic receptors, it is exactly what it sounds like. Norepinephrine is going to receptors for norepinephrine. And again, sympathetic pathways are going to activate and increase the activity of a downstream cell. You may imagine how you can fit all of this onto one sort of board, right? You could take, for example, let me find the image. This image and have your short preganglionic, long postganglionic, and say, okay, these cells right here releasing acetylcholine onto nicotinic receptors, and these cells right here releasing norepinephrine onto adrenergic receptors, for example. So this is a major concept. I would focus a little bit more on how they differ rather than how they are the same. You can maybe imagine how that sacral outflow is in contention right now. You can say to yourself, okay, these two pathways look identical. Maybe this is why we confused sacral outflow for parasympathetic for so long, because they're releasing the same substances onto the same receptors. The other difference here is the target organs. So most body structures that are innervated sympathetically will have this pathway, norepinephrine onto adrenergic receptors. Only sweat glands, blood vessels, and skeletal muscle will have muscarinic receptors receiving acetylcholine. Acetylcholine can be excitatory or inhibitory. This is going to be significant when we get to cardiovascular next semester. So acetylcholine released from sympathetic axons is always excitatory. Most postganglionic sympathetic axons release norepinephrine onto the effector. And again, norepinephrine can have excitatory or inhibitory effects. The difference in effect is what kind of receptor is there. There's actually more than one kind of norepinephrine receptor, for example. All right, you do need to know a little bit about what's going on here. It is on your worksheet for the last week. People often ask me, what's the difference between norepinephrine and epinephrine? And largely the difference is what kind of receptors they attach to, in what regions, and what effects they have. Overall, it's going to be sympathetic activation, right? If you look at the effects here, almost all of them are going to be about fight or flight in one way or another. Vasoconstriction in certain regions, like the gut, where we do not need to be sending a lot of oxygen when we're running away from a bear. We decrease digestion by decreasing blood flow to the gut. But in other places, we're going to need to vasodilate. So for the heart needs extra oxygenation, or the bronchioles of lungs need to widen. Vasodilation in skeletal muscle arterioles get blood into those skeletal muscles so that blood can pump and you can run away. So on and so forth. These are all sympathetic effects in different locations, and some things will have more sensitivity. Alpha-1 receptors have more sensitivity to norepinephrine than they do to epinephrine. In the gut, norepinephrine. In the heart, norepinephrine. In the heart arterioles, in the lungs, in the liver, in the skeletal muscle, epinephrine is the king. And those are beta-2 receptors. Beta-3 receptors only in adipose tissue, breakdown of fats for use as energy in a crisis. More sensitive to norepinephrine. So yes, this is jazz hands over here. What did beta-3, beta-2, beta-1, alpha-2, alpha-1 receptors do? Where are their locations of effect, and what are the effects? Are they more sensitive to norepinephrine or epinephrine? An agonist is something that increases the action of a substance. An antagonist is something that decreases the action of a neurotransmitter. So, muscarinic receptors receive acetylcholine typically. Muscarine can act as an agonist to increase the activity of acetylcholine, which, remember, in some cases was a depressant effect to decrease the activity. The antagonist is atropine, and that's a drug we can give people to decrease the activity of acetylcholine. Nicotinic receptors receive acetylcholine as well. Nicotine is an agonist. It increases the action of the downstream neuron. Alpha bungarotoxin, I'm not super familiar with. That's a drug we can give people to decrease the activity of these cholinergic neurons, the neurons that release acetylcholine. Alpha adrenergic neurons are sensitive to norepinephrine and epinephrine. Phenylephrine is an agonist that increases the activity of adrenaline, or acts like adrenaline. Alpha blockers, such as Flomax, will decrease the action of adrenaline. And then, not surprisingly, beta blockers decrease the activity of beta adrenergic receptors to decrease the effects of norepinephrine and epinephrine. So the reason this chart is here, it's a few examples. I can't remember if I specifically test on what these drugs are. The idea is to get you into that mindset of pharmacology. This is how pharmacology works. This is how all of our drugs work. All of our drugs work based on agonism and antagonism of receptors. Receptors for neurotransmitters, receptors for hormones. That's how literally every drug works. It's all about receptors. We need receptors, we need proteins on our cells that respond to drugs in order for drugs to work. So recall that everything in your body is going to have dual innervation. You're going to have a nerve from the parasympathetic nervous system going to an effector organ, and you're going to have a nerve from the sympathetic nervous system going to an effector every time. So remember, pupils dilated as a result of sympathetic innervation, pupils constricted as a result of parasympathetic innervation. That means you needed a parasympathetic nerve to get to the muscles of the iris, and you needed a sympathetic nerve to get to the muscles of the iris. They're both always there. Their activity increases or decreases based on your demands, and they're always balancing each other out, so that ideally your pupils should always have the correct degree of constriction for the environment, the light sources nearby, how close things are to your eyes, and how fight or flighty or resty digesty you are. So this is, you could be running from a bear or you're just in low light. Either way, your sympathetic nervous system caused that. It's always a balance. It's always working. You don't have to be running from a snake for your sympathetic nervous system to activate. You just have to have a need that is innervated by the sympathetic nervous system. It's all homeostatic mechanisms. You always have homeostasis working in dynamic balance to control your internal environment to give you the appropriate response to your environment. So these occur reflexively. You don't need any conscious knowledge of it. Some autonomic reflex examples: your bladder gets full and you have sensory receptors for stretch in the lining of your bladder. That goes to your spinal cord and innervates an effector that is going to cause involuntary contraction of smooth muscle around the bladder, involuntary relaxation of sphincters that hold your ureters closed, and that is going to lead to urination. Luckily, you've got a voluntary muscle as well, so you can still hold your pee once this has taken course. But the urge to pee is based on an autonomic reflex, and then you have a voluntary component so that you can control it before you get to a restroom. Ideally. Obviously, that can break down in some people. It is pathological if that no longer works. It is never normal to have urinary incontinence. Your autonomic nervous system is influenced by your cerebrum, your higher order thinking, your knowledge of where you are, your, oh my gosh, I just saw a bear. Your hypothalamus is in control of all of your hormones in your body, pretty much. And it has a lot of receptors and sensors for the chemical state of your body, so that's going to moderate your autonomic nervous system. Your brainstem, and that just says spinal cord, I don't know why that still says that. Your brainstem and spinal cord. Your brainstem because it contains a lot of your cell bodies for your cranial nerves, and also because it has a lot of your sensory and motor functions embedded in it, and same for spinal cord. Sensory and motor pathways, pathways for all of your autonomic, sympathetic, parasympathetic, voluntary, involuntary, everything goes through the brainstem and spinal cord. Cerebrum, conscious activity, hypothalamus, integration, command center, brainstem, major reflex centers, spinal cord, reflex centers, especially for defecation and urination. That is it. We have finished. Only took an hour. I'm sure there will be parts that you'll need to see once or twice. You can and should try to fit this all onto one piece of paper. It is doable. All of the significant stuff, all the stuff that you'll actually carry forward with you into ANP2 and pathophysiology fits on one page. So I would recommend that page start with something like this. Start with craniosacral, parasympathetic, thoracolumbar, sympathetic, cranial nerves, 3, 7, 9, 10, splanchnic nerves down here, cell bodies from the lateral gray horn here, preganglionic short, postganglionic long, preganglionic acetylcholine onto nicotinic, postganglionic norepinephrine onto adrenergic. Then for your craniosacral division, preganglionic long, postganglionic short, preganglionic acetylcholine onto nicotinic, postganglionic acetylcholine onto muscarinic. And then list some effects on different organs. So sympathetic division, increased heart rate, increased pupil diameter, increased bronchial diameter, so decreased bronchial constriction, decreased blood flow and activity of the viscera. Parasympathetic division, salivation, lacrimation. Let's see, what else we got? Decreased heart rate, decreased bronchodilation, decreased bronchial diameter, increased blood flow to the viscera, increased activity of the digestive organs. So here's a fun thing that kind of only works before we figured out that sacral outflow is sympathetic as well. And it makes me really sad that I can't officially use it anymore, but for our purposes you can. Erection is considered a parasympathetic activity. Ejaculation has been considered a sympathetic activity. And the way some people remember that is by saying point and shoot. Point standing for parasympathetic and also being symbolic for an erection, and shoot standing for sympathetic and being symbolic for ejaculation. So until we discovered this, it was different things affecting different parts of that reproductive cycle, of that coitus. And we don't get to really say that anymore, which makes me super sad because it's point and shoot. It's hilarious. No, can't do it anymore. Womp womp. Alright, I think that was a pretty good review and summary. Also, in addition to sympathetic being noradrenergic, do a little bit extra focus on this chart. Beta 3 receptors, only adipose tissue, only lipolysis, more sensitive to norepinephrine, so on and so forth. Our significant, if I had to say most significant stuff here, I would say adipose and maybe heart and maybe guts is what I would focus on. But I do have it on your worksheet, so you will get some more experience with that. Okay, let me know if you have any questions. I will put this up early and post it so that you can ask questions on our last day of class, whether that's a review day or during week 10 as usual. Let me know if you need anything.